So Olga is an accomplished biopharmaceutical scientist with over 2 decades of industry experience, specializing in cell line development, mammalian protein expression, and biologics manufacturing. Her extensive career spans multiple leading organizations, including Mural Oncology, Alchemers, Cell Signaling Technology, Bluefin Bio, and Patheon. As a PhD scientist, Olga has established herself as a technical leader in translating research concepts into robust clinical stage drug substance production workflows. While her work appears to be primarily focused on industry applications, her expertise in biopharmaceutical development processes has contributed significantly to advancing biological therapeutics from research through manufacturing stages. With that, I'd like to welcome Olga.
Thank you, Amit, for introduction, and thank you everyone for attending my talk today, where I'm going to talk about how we accelerated cell line and process development using the leap-in transposase expression platform in application for the molecule. So one of the companies I work, it was a cytokine-based company, and we were developing novel cytokines to treat cancer patients. And the molecule lead sequence we received from research group was an open-hole Fc scaffold. And the long-term goal for this project was to develop a high-yield cell line and scalable process within a tight IND-enabling timeline. And this goal came with several challenges. Some of them was we observed low expression level during transient transfection, probably caused by open-hole heterodimer complexity. And also it was our first internal project. We did not have any system in-house we like. So to overcome these challenges, we came up with this vendor execution strategy to evaluate 10 expression systems available on the market, then to select 3 and bring them for in-house evaluation at the pool stage, and then select 1 and start cell line development. For the critical assessment of vendors, as you may know, there are so many vendors available on the market. And in our situation as a small company, we were looking for a vendor who can offer a platform feed, cells, media, off-the-shelf, use off-the-shelf materials, and also vectors. Next generation productivity, evaluation cost was important, and also, most importantly, future licensing fee and structure. Because we need to understand how much do we need to pay later on as IND, BLA, and royalty fee. I was looking for a system also which has already molecules in clinic, and also expertise and customer support was important in this evaluation. So based on this criteria, we selected 3 expression systems, OGS-based, for in-house evaluation at the pool stage. So system A and B, it was novel transposase-based technology with a semi-targeted integration, and system C, it was an inducible expression system. I had a very good relationship with this vendor, and they recently established a system for Frankenstein molecules. And they already have some product in the clinic. So next step was to evaluate the system in a pool stage and select 1 for clinical and manufacturing. Time-wise, we had 6 months to bring systems in-house and evaluate at the pool stage, and then to deliver the final clone within a year. Here it is our cell line development workflow. As you may know, the process usually starts with selecting the lead sequence or sequences, then which will be used for, then we need to create expression vectors, which will be used for transfection and pool selection, followed by single cell cloning and clone screening. Then research cell bank creation, phenotypic stability, genetic characterization, and then lead, clone, and probably backup will be selected based on growth, productivity, product quality, monoclonality, stability. Then prior to create master cell bank creation, we need to evaluate clones for safety. They need to be free of any bacterial and viral contamination. Then master cell bank or working cell bank will be used for manufacturing. So this is a cell line development workflow, and we know that it's not a linear process, and many stages are overlapped and required a lot of interplay between department and functions. When we were bringing systems for in-house evaluation, there is a lot of time when you work with the legal teams on assigning service non-disclosure agreements and material transfer agreement. And also we work with the protein therapeutics team, selecting the lead sequence, and also trying to understand the critical quality attribute for the molecule. Later on, there is a lot of interplay between upstream, downstream, and analytical departments. And when we are getting ready to move to the manufacturing, we also work closely with the technical operations, quality, and regulatory. So here it's our manufacturing cell line development workflow. And next, I'd like to focus on evaluation of 3 expression systems, which we selected, ABC. So the next step, we need to create expression vectors. For the system A, we went to this comprehensive approach, and we generated multiple vectors, single gene vectors, double gene vectors, and also we play with the GS stringency, different promoters, and positions of gene of interest in the vector. With the system B and C, we did not have a lot of budget and resources. We went with a less comprehensive approach and created 2 vectors, one for knob arm and one for a whole arm, with a goal to create at least 2 stable pools with a different co-transfection ratio, 2:1 and 3:1, knob and a whole. Why this ratio? It's been shown during transient expression using different systems that it will be beneficial for productivity and heterodimer content. So next, we perform transfection, which we use electroporation and pool selection. And also GFP, control antibody, MAC, no DNA control, was used for these experiments. Pool recovery. So transfection and pool recovery for system A were performed according to the vendor protocol, and total of 9 stable pools expressing the target molecule were created for further evaluation at this stage. You can also see that all pools have successfully recovered in 18 days. We did not observe impact of vector architecture on pool recovery. For the system B, it was slightly different situation. Vendor did not provide us a BioRat electroporation protocol, and it was only electroporation device I had in-house, and I used just 2 post-transfection recovery methods. So expression pool 12 and 13 with a different co-transfection ratio, as well as GFP, control antibody, and no DNA were created using method 1, and pool 14 and 15 with a different co-transfection ratio created using method 2. If you look at the viability profile, it's a little bit busy, but basically mock pool cells did not recover it. It was expected. That pool 12 and 13 created method 1 had a very slow recovery, and according to the vector manual, they told us if we observe this low viability, we need to discard the pool and start it over. But because I already have good recovery in pools 14 and 15 with the same molecule and co-transfection ratio, I decided to disregard pool 13 and keep pool 12 just to see if it will affect the productivity and product quality. So a total of 3 stable pools expressing our target molecules were moved for further evaluation. For the system C, unfortunately, we did not create any pools. And because we had a significant delay in the material transfer agreement, and the lesson we learned that it was a government organization, when you work with government organization, sometimes you need to allow yourself longer activity to sign these legal agreements. So from now on, I will be focusing on the pools of system A and B. Next, after we created the stable pools, we evaluated them in a 14-day fed batch assay in a shake flasks for cell growth and productivity. Here it is a productivity profile of 9 pools expressing the target molecule created using system A in a different vector architecture, which I listed previously. And clearly you can see that productivity-wise, some of the pools produced more than another. If you look at pool 4, it produced 3 times less material than pool 8. However, when we look at the productivity of the 3 pools created system B, they all showed us superior productivity. And at this stage, so we were very happy with the result, and at this stage, all eyes were turned into the system B pools. And for us, next, it was important to understand if we are producing the right heterodimer, the right form of material. So these 3 pools were 1 step purified using proteinate chromatography and only 1 eluate from 1 fraction was analyzed for purity and mass spec analysis. And when purity results came back, we were able to see that all purity assessed by size exclusion, only 1 fraction, were acceptable in the pool stage in a 1 fraction. Also, I'd like to mention that pool 12 had a higher purity, that low viability during the pool recovery did not impact this pool. Next step, importantly for us, was understand the heterodimer mass, and when we ran a mass spec analysis, we observed expected mass for all evaluated pools. So based on these critical quality attributes, we decided that all pools were acceptable to move forward. And this was our turning point in our program, because based on the data, team just saw the opportunity to advance this project further. We already selected the expression system, definitely system B, and next step, we just proceed with a single cell cloning, upstream, downstream, and analytical process development. With the next long-term goal or final goal to execute 50-liter bioreactor run and generate material for the upcoming GLP toxicology study, then to select the final clone and create master cell bank creation, and also we start working on technology transfer to manufacturing. This goal also came with challenges as a strict timeline and limited resources. Strict timeline, we already scheduled the slot for toxicology study and master cell bank creation. Limited resources, we still were a small team, and we had also in a parallel late-stage project, and many resources were focused on it. So we needed to be mindful. Next, we use single-cell cloning workflow, and for this workflow, we use only pool 14 and 15 for single-cell cloning created using co-transfection ratio. And then total of single clones, 100 single clones from each pool were screened for productivity in a microwell plate format. Then based on productivity only, I didn't go deeper in who is growing faster, who is not. I selected 26 clones from each pool. for scale-up and further evaluation in Amber 15, which I will tell you in the next slide about our upstream development plan. So our upstream process development plan was running in parallel, and we decided firstly to execute process parameter screen in Amber 15 using one pool to identify optimal process condition, which we can then apply to 2-liter bioreactor run, from which we can generate material for downstream and early analytical development, and finally to execute a 50-liter pilot run to show scalability and, importantly, to generate material for the upcoming toxicology study. For the Amber screen, for first Amber run, we utilize this small DOE methodology where we trying to understand effect of pH, seeding density, and temperature set points on and the impact on the growth and productivity using only one pool as a model. And surprisingly or not, but all conditions showed us a robust cell culture performance and protein expression level. So here it is we ran 24 stations in duplicates, and titer-wise, we saw the identical almost level, and we just choose one condition, which we applied for subsequent clone screening. Which came next, because while we were evaluating pools and Ambers, our clones created from a single-cell cloning workflow were ready to be screened in the next Amber runs. And total of 48 single clones were screened in next Amber runs for cell growth and productivity at first. Here it is a productivity profiles of these clones. And at this stage, because we were ahead of our purification and analytical team, we were thinking how we can distinguish product quality from all 48 cultures prior to purification. And we came up with this conservative approach where we analyze when we ran SDS page gels on the supernatants because we have enough product, and we were able to distinguish clear clones from more clones with a more purity. So based on these results, combining pool origin in the cell line development, usually we prefer to select clones from at least 2 different pools. Cell growth profiles, I'm not showing it here, productivity, monoclonality, and SDS pH results, we selected top 4 clones. And next step was to create research cell bank, and this research cell bank was used for stability study, genetic characterization, 2-liter bioreactor runs, purification, and product quality analysis. For stability study, one of the industry approach, we are pathogen cells for 6th generation with and without selective pressure. Then research cell bank and research cell bank plus 6th generation, we will analyze in a 14-day feed batch assay. So all 4 clones, here I'm showing results only for 1 clone, but all 4 clones met the industry stability requirement, higher than 70% of consistency in the original growth and productivity. And actually, it was 100%, I will say, of stability, which is not a surprise for transposase-based system. In a parallel, we ran 2-liter bioreactor for 4 lead clones and 2 associated pools, and we were able to see the consistency between cell growth and some parameters. Only 1 clone showed us a drop in viability, around 50% at the end, and the productivity-wise, we were able to get between 6 to 8 grams per liter for all the cultures. So these cultures were purified, and the next important criteria for us was to understand the glycosylation profile and compare it with the reference material which was generated during different transient expression system. And when we ran this analysis, we were able to see that all glycan species were present among all the cultures, supporting consistency in post-translation modification. The next important criteria was genetic characterization. It was important to understand that the vector we put into cells still are here. So we use Sergentis and the targeted locuses amplification technology, and we genetically characterize cells, vectors, and we were able to see that 2 out of 4 clones had a single point mutation in the vectors. And the number of integration sites was different, meaning that our clones were different. And then we also characterized research cell bank plus 60 generation only for 2 top clones, and we did not see any mutations, and integration sites maintain the same. So combining this data with the results from 2-liter bioreactor run, it was easy for us to select the top clone based on a harvest titer and the backup clone without a mutation. So top clone was selected, and next step was to generate toxicology material for upcoming toxicology study. At this stage, when we performed the vial saw, we still didn't know which culture we will use. Are we going to use top clones or mixture of clone or one clone? And the genetic characterization data came back at this stage, and we decided we will just use one lead clone for toxicology material generation. Lastly, one of the last slides, I just want to show the robust cell growth and productivity for the final clone for system generated across the scale. You can see 50-liter bioreactor run, Amber and 2-liter bioreactor run showed us a comparability between each other. So it was very robust. And using this approach, we were able to accelerate our original timeline, and in 6 months, we already delivered the final clone, initiated master cell bank creation, and around here, I was already in a manufacturing site overseeing the master cell bank vial saw. And this is my final slide, a summary takeaway that the successful delivery of this project was not only a technical win, but also the example how important is to select not only expression vector, but expression system, and apply automation, be meaningful to your resources, and use a teamwork to deliver a project. Thank you so much for your time, and I will be happy to answer any questions.
Thank you, Dr. Rimkevich. Really appreciate your talk.
Hi, thank you for the nice talk. I know that Adam is good at making vectors, so I want to ask your opinion on constructing vector for bispecific antibodies. Do you use 2 different promoters for knob and hole or different chains?
So if I understand you correctly, you ask if I am using different promoters. So this was a proprietary system. I cannot disclose this data, but we try different promoters with the system A, yes.
So that's basically a difference of stringency?
Yes, we also try different GA stringency and different promoters.
So you just, the last question is, do you control the ratio based on the concentration of the 2 different vectors, or are you using one single vector but different, like,
so when we design double vector with an open hole, we control this ratio. I don't know if it's beneficial, but for the co-transfection, we use 2:1 or 3:1 ratio. Our protein therapeutics team used 4:1 and 1:1, and they made the conclusion that this is beneficial.
Thank you.
Maybe I'll ask one question. So one of the top 4 clones, first of all, I'm always amazed by transposase based and especially atoms technology with the consistency. What we get, one of the top 4 clones showed reduction. I think it was on slide 25, and then your team did genetic characterization. I wonder if there was any copy number also evaluated, or if the team also identified why that clone showed drop in viability.
So we did not evaluate any copy numbers. We analyze integration sites. Sometimes people think it's a correlation. But we were just we were moving fast with this project, and for us it was not important to understand the copy number at this stage. So we don't have this data, and also we were trying to save a budget for a company.
Yes.
Thank you, wonderful talk. So your pool titer versus clone titers are not much different. So it's very, very predictable, the clone titers from the pools. Have you done stability study on the pools?
No, we did not do. We are not doing stability study on the pools. Yes, and some of the people who are presenting this transposase-based results, technology, they see the increase in the titer. And actually for the system A, for one of the pools, we performed single-cell cloning with the hope that clones will produce more material. But it was, again, pool was the average, and we did not see a big improvement of productivity.
Just wondering, like if pool material can be used with so much of high productivity, if we can skip clone production and use pools for material generation, if you can show pools being very stable.
Yes, we can, and we were considering to use pools for GLP toxicology study, material generated from pools. But we were a small company, it's our important project, we just decided to be on a safe note.
Yeah. Thank you.
Any more questions? Okay, if not, let's thank our speaker once again.